Alrighty, hey everyone, how are you? My name is James Riley Jacobson, and I'm doing my presentation on the topic that I picked, which is the evolution of the turtle and its shell. All right, just to start off, a little introduction. So the turtles come from the order of the Testudines. So throughout this presentation, I'm definitely going to be referring to sort of the entire order and all of turtles in general as Testudines. So just some fun facts. There are about 313 different species of turtles with 143 subspecies. And out of that makes a total of 456 turtle taxa. So quite a diverse organism. Some other facts that the fossil records show that the oldest common ancestor of turtles lived about 200 million years ago. So a fairly old and long phylogeny to go along with this organism as well. Alrighty, according to the PowerPoint, they are amniotes, which means they do lay eggs and they do breathe oxygen. They have a very special shell made of sometimes referred to as dermal armor, and it's definitely special and unique to the turtles in that other kinds of organisms, for example, such as armadillos or anything with a hard carapace, they don't come from the same common ancestor. They're all different in the kinesis and formation and development of their shell. So turtles are split into aquatic and terrestrial. So if I ever say turtle, you can be sure that I'm talking about aquatic turtles, either those turtles that reside mainly in rivers or lakes or any kind of let's see, body of water or the ocean. Those will be referred to as turtles. And then if for terrestrial of the order Testudines, they'll be referred to as tortoises. Okay. Something that's important to understand is that the oldest common ancestor of turtles of the order Testudines was in fact aquatic, and then afterwards that aquatic species actually died out after giving rise to several terrestrial species. And then funny enough, from those terrestrial species came new aquatic species, and eventually those terrestrial species died out, and then from the aquatic species that survived after that third extinction, that third branching off in the phylogeny, that's where we have the aquatic turtles that we have today. So as you can see, the terrestrial and aquatic, it's a sort of homoplasy that kind of went on and off. We didn't have, not all the turtles came from the same aquatic ancestor and remained aquatic. You had all the aquatics dying out and then new aquatic ancestors coming from terrestrial turtles. Alrighty, so for turtle families, there are 14 turtle families. 11 fall under the subfamily of Cryptodira, and 3 fall under Pleurodira. Something that characterizes these different suborders is the Cryptodira known as the hidden neck turtles, and the Pleurodira are known as the side neck turtles. So the hidden neck turtles are characterized by being able to completely lower their heads and retract them fully back into their shell. And the side neck turtles cannot quite fully retract their necks, which leaves some of it exposed. So it's pretty self-explanatory by the names. So I'm just going to name off all these families real quick. I'll give the Latin name that they have, and then I'll kind of describe what they are fairly quickly, so as to not spend too much time on this. So you're going to have your Ceratochellidae, which are your pig-nosed turtles, your Chelonidae, which are your sea turtles, your Chelaridae, which are your snapping turtles, your Dermatimidae, which are your river turtles, your Domochilidae, which are your leatherback turtles, Emididae, your pond turtles, your Geomyidae, Asian box turtle, your Cynosternidae, which are your mud turtles, your Plesternidae, which are your big-headed turtles, and your Testunidae, which are your tortoises. And then your Trionchidae, which are your soft-celled turtles. So those are the 11 that fall under Cryptodira, the hidden neck turtles. And then there's 3 more families that fall under the side neck, Pleurodira. And that's your American side neck turtles, your African side neck turtles, and your South American side neck turtles. All right, so there have been a lot of origin hypotheses. The turtle is pretty mysterious, and it was really hard through finding fossils. We were never able to find enough fossils to completely understand what what were the intermediates between the most common ancestor of turtles and the modern day turtles that we have today. So because of that, it was fairly difficult to know if the common ancestor that we thought was for the turtles was the actual common ancestor. So they split it down to Parasaurs and Procolophonids. The Parasaurs were land-based and the Procolophonids were aquatic reptiles, aquatic dinosaurs. And between these two, they could never really figure out which one was the common ancestor. And one of the reasons that they didn't trust this is because of a lot of the flaws in the phylogeny based off of supposed morphological homologies. And the example that I'm going to use for that is the dome shell versus the flat shell. So the original hypothesis that was used to make phylogenies and separate turtles into different families and groups was that if the turtle had a dome shell, it was thought to be terrestrial, and if the turtle had a flat shell, it was thought to be aquatic. But we found that there's actually a flaw to this. So that turtle that you actually see in the picture has a very flat shell, and that's actually the African pancake tortoise. And as you can see, it's definitely not aquatic, it's a tortoise. And then, for example, for the domed shell, the aquatic Asian box turtle has a very, very domed shell. So as you can see that we can't really rely on these physical homologies to actually be able to group these turtles. So kind of had a difficulty there. So I'm going to go through some of the physical homologies, some of the physical homologies of turtles that are very unique to turtles, and classifying them and classifying related species and ancestors is the dermal armor. So the dermal armor, this composes the shell of the turtle, and it's formed from the vertebral column and its ribs, as you can see in the picture. So it's kind of on the back. So I'll just explain this picture real quick. A is going to be your plastron, which is the bottom side of the turtle. B is a posterior view, and that's your carapace. And then C is an anterior view, and you can see the ensnarement of the ribs in the back, posterior, right behind, posterior to the pectoral girdle, which is the acromion process, which is labeled here in the thing, which is labeled here in the PowerPoint, and along with its scapula. So to explain a little bit more of the homologies, it's really important. The acromion process is a really important homology in distinguishing between a lot of the other hypothesized ancestors, and that is because the acromion process of the turtles is pretty unique in its location in relation to the clavicle of the exoskeleton. It's underneath the clavicle. It's kind of over the clavicle rather than underneath the clavicle, as you would see in other mammals, for example, us as humans. The astrologocalcaneal complex is another fairly important complex. It's formed from the fusion of the calcaneus, talus, and tarsal bones of the foot. And this complex is present in other commonly mistaken ancestors, such as the parasaur reptiles. Let's see. Both species contain this complex, but each progress a little different in the final structure in adulthood. And that's one of the reasons that it's kind of hard to know what is whether the parasaurs really are common ancestors, because you see that different. Another important homology that you're going to see is that hooked fifth metatarsal bone in the foot. And that is going to be it for the homologies. The only last thing I can say is the terrestrial short foot versus the aquatic long foot. Your terrestrial turtles are going to have short feet. Are going to have shorter foot, while the aquatic is going to have a longer foot. And that is a physical homology that is very characteristic, that there will be no mistake. If there is a long foot, the turtle will most likely for sure be aquatic. All right, I'm going to talk a little bit about the molecular phylogenetics. Molecular phylogenetics were pretty important before we could fill all the fossils and fill the fossil record and actually have intermediates. Before we could find all of the fossils to make that phylogeny, we had to rely pretty regularly on molecular phylogenetics. So a lot of the case studies and the tests that I was able to research involved the RAG1 gene, or the nuclear recombination activase gene, and the RAG1 is also involved with RAG2. And what these do is these actually work in, these are genes that, not genes, they're proteins that work to take genes that work in our immune system and and make antibodies and proteins, and we're able to, these RAG1 and RAG2 are involved in cutting out DNA out of these and modifying the RNA from these genes to create the correct mRNA template to be able to make these antibodies. So they were able to study this RAG1 because it was pretty common and it was very specific to each species. And they were actually able to fix the phylogeny a bit. So as you can see, the Chiluridae, the snapping turtle, and the Plastostyrinae, big-headed turtle, these two species were thought to be fairly close, but it actually turns out that they're pretty different. They're pretty different according to molecular phylogenetics, and that's just an example of what it did. And then through molecular phylogenetics, we were kind of able to know the branching of the turtle, of the family, and we were able to work to be able to understand how this branching occurs. So out of the families that I had spoken with you about, the first branching that we would see occur would be with the Cheluridae family, followed by the Dermochelidae family, and then the branching would go to the Kinosternidae family, the Amidae family, the Plastostyrinae family, the Geomididae family, the Testudinae family, the Dermochelidae family, and then the last branch would be the Chelonidae family, in that order. So that's what molecular phylogenetics did for the turtle, is that we were able to figure out the order. So there is definitely a large variation in size for turtles. As you can see, you've got the leatherback sea turtle, which is just absolutely enormous. Another example would be the Galapagos tortoise, which is the largest known species of the Testudines. And then as you can see right there to the right, we've got a little tiny turtle. Okay, so the smallest known turtle we have is 80 grams, and the largest is 417 kilograms. Based off of studies and based off of what scientists have been able to figure out based off of certain evolutionary novelties, we were able to figure out that the size is based off of habitat. So this could be lack of predation. This could be due to trying to survive. There's definitely a larger fitness for the larger bodies, and that's why in certain habitats we started seeing these large turtles coming about. Another reason was for preparation for colonization. So we believe that these certain turtles grew in size to prepare themselves for colonization and for long travels. Obviously, they need to have longer sizes to be able to evade predators and to be able to have the energy to travel long distance and be able to last during certain conditions. And what we believe is that after colonization was over, they would have shrunk back in size. But as you can see, for example, the Galapagos tortoise, they stayed fairly large. As I said, the habitats and the new niches that they eventually occupied, there was a lack of predators and there were a lot of other things that accounted for, such as an increase in resources and plenty of food for them to keep their size. So the turtle shell shares several functions other than protection, as I wrote there. Some of the other key purposes is as a reservoir for water and also a pH buffer. So the shell is unique in that it's made of several different structures that will eventually all fuse together. It's made of 59 bones and 54 scales. So the shell kinesis is quite unique in that all the dermal bones of the carapace are fused together, and each individual bone binds to the dorsal surface of the costal ribs. It is the distal ends of the costal ribs that eventually will connect to the peripheral bones, and this is made by the development and fusion of two key structures, which is the epiblast, which is homologous to the clavicle bone, which forms the superior ventral border of the rib, and the endoplastron, which is homologous to the interclavicle bone and forms the medial portion of the plastron, which is the anterior or the bottom side of the turtle shell. The two most important stages in development of the turtle shell would be the ensnarement of the ribs and the lateral growth of the dermis, which is the top portion of the carapace, or the original, or the starting point for the carapace. It is a carapacial ridge that is key in these stages and key to the formation of the turtle shell. So this ridge is what defines turtles from other reptiles, especially during the embryonic stages of life. That's how we can really tell the difference and the uniqueness of the turtle shell from other species that have some kind of dermal armor. The ridge causes the lateral growth of the dermis, and that's what gives the turtle shell that distinct development, or that distinct structure. And then with the lateral growth, the ribs usually follow, and that is the ensnarement of the ribs that I was talking about. And they follow the dermis until they fuse with the peripheral bones right there on the lateral edges of the shell, and they eventually fuse with the plastron, or the interior view of the shell. So here's your actual origin discovered for the turtles. So as I said, we had to use molecular phylogenetics at first to be able to make a lot of connections, but even then with molecular phylogenetics, we still lacked information about the intermediates and how that turtle shell and dermal armor eventually formed. So the final intermediate species that we were able to find to complete the phylogeny was the discovery of the Odontochelys semitestacea, which I probably botched that name, but that is it. And it was very important that we finally discovered this, because we were finally able to link it to the aquatic-based ancestor that I had spoken of earlier in this presentation. And we were finally able to make that link, and we were finally able to understand more fully how the dermal armor, or the turtle shell, finally developed, and what species were involved, and what species are related to the order Testudines. So I actually have them all listed here in order. So you have the lizard family Milleretta, that was first, which eventually evolved into a family lizard known as Unitosaurus. Eventually came the family of lizard turtles, which is Odontochelys, which was discovered recently, which finally made that connection. We finally understood when we finally discovered that intermediate species that resembled lizards as well as turtles, and finally connected the two species. And then we have our first identified turtles, which is the Progonochelys. So in conclusion, to wrap up, I just went and found some fun facts about turtles. So because there's several turtles that are migratory, they're always in danger from fishing. Hundreds of thousands of turtles are dying constantly throughout the year, and it's definitely a big deal, and definitely something to be aware of. We have many species have gone extinct due to human involvement, due to lack of laws protecting the turtles and lack of laws protecting the environment. So definitely something important to understand, that there are several species that have gone extinct, and that there are several species that are still in danger even today, that are close to being extinct. Something to understand is that turtles are an R-selected species, which means they produce a large number of offspring due to high levels of predation. So once again, they're already used to high levels of predation. Human involvement only makes things a little harder. And obviously, some of the things that we can do is to increase the awareness of this, and to help to pass more laws to protect these amazing creatures, and this amazing example of evolution, because turtles truly are a novelty of evolution. There aren't many organisms that are like turtles. They are very unique. And this is my presentation, and here are my references. If you have any questions, just write in the discussion board or write me, and I can try to answer it the best I can. Thank you. Bye.